Good evening, everyone, and welcome to UT Southwestern Science Cafe. We welcome back both our regulars and also our new guests to this ongoing series. My name is Jenny King, and I'm part of the public affairs team here at UT Southwestern. This is our third year to host Science Cafe, and we are proud to produce this series. On behalf of my colleagues, Charlie Thompson and Joya Lang, as well as our guest speakers, Dr. Maria Cha Rohr and Dr. Peter Tsai, thank you for joining us tonight. Science Cafes are online conversations where our speakers take you on deep dives into science and healthcare topics. At UT Southwestern, we are an academic medical center, and that means we bring together the research, medical and health education, and patient care all into one institution. Research and education informs care advancements quickly, which is a benefit to our patients and our community. We have fun at Science Cafes while we learn and we engage during the program. We are also respectful of all perspectives, especially with regard to points of view about autism diagnosis and treatment. And we have two of our best here tonight. This evening, we're discussing autism spectrum disorder, the pathway to progress, with Dr. Maria Cha Rohr and Dr. Peter Tsai, and more of them in just a moment. Before we get started, we have just a couple technical points to cover. We are recording tonight's program as well as live streaming it on our UT Southwestern Twitter page. We want everyone to mute your microphones to help with audio clarity for the whole program. We encourage you to use the chat feature to list questions for both doctors. We're monitoring your questions in the chat box, and we will start Q&A at the conclusion of their presentations. Charlie will be facilitating this part of the program. And finally, while we cannot answer personal medical questions, we would love to hear from you with your general questions about autism. And with that, I'm so pleased to introduce our guest speakers. Dr. Maria Cha Rohr is an associate professor in the Eugene McDermott Center for Human Growth and Development and the Center for Genetics of Host Defense, and in the departments of neuroscience and psychiatry. She obtained her PhD in molecular and human genetics from Baylor College of Medicine, where she studied molecular mechanisms underlying Rett syndrome. Dr. Cha Rohr is the principal investigator of her lab. Dr. Peter Tsai is an associate professor in the departments of neurology, neuroscience, pediatrics, and psychiatry. He received his MD and PhD from the David Geffen School of Medicine at UCLA. Clinically, Dr. Tsai cares for children with autism and cerebellar dysfunction in the Center for Autism and Developmental Disabilities, and he is the principal investigator of his lab. Charlie is pasting both of their bio links in the chat. We are starting our presentations tonight with Dr. Cha Rohr, followed by Dr. Tsai. Dr. Cha Rohr and Dr. Tsai, thank you so much for being part of Science Cafe. Dr. Cha Rohr, the virtual podium is yours.
Thank you, Jenny. Hello, everyone, and thank you for joining us for the Science Cafe. We're really excited to be here. And tonight, I'm going to talk about the latest advances in autism genetics and the study of disease mechanisms, and how this information can help us think about targeted therapies for some forms of autism. But before I dive into that, I just want to thank the individuals with autism and their family members who participate in our studies and who drive our research. We are grateful for their contribution to advancing our collective knowledge of autism. I want to start first by defining how we think about autism. Autism is a spectrum of neurodevelopmental conditions characterized by impaired communication skills, social behavior abnormalities, restricted interests, and repetitive behaviors. It affects 2.3% of children in the United States, or about 1 in 44, with males being more affected than females, on average about three times more. In addition to the core symptoms that I mentioned, there are other conditions that are also frequently seen with autism. And some of these, what we call comorbidities, can be medical, developmental, behavioral, or psychiatric. Some examples include GI abnormalities, intellectual disability, motor function abnormalities, anxiety, and seizures. Because autism is a wide spectrum, and we have a quick poll question here about autism diagnosis. So I'm going to continue as everyone answers this. Because autism is a wide spectrum, Every individual is very unique. So if you've ever met an individual with autism, you've really only met that individual with autism. And in our endeavor to better understand this condition, we want to be inclusive of the full spectrum. And that includes individuals with mild symptoms, as well as individuals who have more severe symptoms with significant impact on quality of life. So I told you about how complex autism is in the way that it presents clinically. But what causes autism? We know from many studies that autism is highly heritable. And what that means is that changes in your genes account for whether you have autism or not. And work from many research teams has identified hundreds of ASD genes that, when altered, give rise to ASD. And a proportion of these genes are associated with well-defined syndromes such as Fragile X, Rett, TSC, or Angelman. And another proportion of ASD cases are due to changes in big parts of DNA. And we call these chromosomal abnormalities or copy number variants. Another percentage is due to de novo or spontaneous point mutations. And another percentage are due to inherited point mutations. And finally, some cases are due to somatic mutations, and these are ones that only occur in the brain. For the majority of cases, about 65% or so, the underlying genetic causes have not yet been identified. And I want to point out that all of these variants are ones that are very rare in the population. And over the past 2 decades as a field, we've made significant advances in Asd genetics. Can everyone see my screen, or do you see the poll when it pops up? Okay, great. So over the past 2 decades as a field, we've made significant advances in Asd genetics. And although hundreds of genes have been identified, each of these genes on its own contributes to a very small proportions of individuals with ASD. Okay, so because of all the data from human genetic studies to date, we can start thinking of ASD as a collection of individually rare genetic subtypes. And in order for us to start understanding the pathophysiology of ASD, I would argue that we have to understand the molecular mechanisms underlying each of these different genetic subtypes so that we can begin grouping them based on specific pathways that are disrupted, and we can start thinking about therapies, but also more importantly, to inform diagnosis. Of course, thinking of ASD as a collection of individual genetic subtypes doesn't mean that you should think of therapies for one gene at a time exclusively. You may need to do that for some genetic forms, but for others, you may be able to combine the subtypes based on deficits in a common brain circuit or pathway, or even an affected brain region. When thinking about therapies, the ideal path may appear linear, going from gene identification to therapy development via animal models. However, for complex disorders, and particularly ASD, the genetic and phenotypic heterogeneity that I just told you about complicate this path to therapies. And possible therapies will need to be customized based, for instance, on the specific underlying molecular changes in these different genetic subtypes. My lab here at UT Southwestern is interested in identifying ASD genes, studying their function in the brain and the molecular mechanisms that they regulate, so that we can use this information in diagnostics and the design of.
Dr. Cha Rohr, I muted you. Can you unmute yourself? Sorry.
Yes. Okay. So the focus of our studies are the more severe genetic subtypes with debilitating comorbidities. And now I'll tell you about some of our ongoing studies and how we identify ASD genes and investigate their function. Okay. We identify ASD genes through genomic sequencing in families with ASD. We have protocols that are approved by the UT Southwestern Institutional Review Board, or IRB, to recruit and enroll participants and perform these studies. We partner with our community and the families to include samples from ancestrally diverse participants and participants across the ASD spectrum. And we collaborate with our clinical colleagues to identify interested study participants and obtain detailed clinical information. Our goal is to identify ASD genes and ultimately to inform diagnostics and therapeutics, and in doing so, to learn about normal brain development, social behavior, and language. The Asd Genetics cohort that we've enrolled so far includes many ancestries, shown here on the left in percentages, and the plot on the right shows how samples from this cohort, which are shown in bright yellow, are spread across populations from all ancestries. We are really excited about this because to date, all of the human genetic studies in ASD have focused on cohorts that are predominantly of white European ancestry. And we're looking forward to the discoveries that we'll make in our ancestrally diverse cohort. So now, you have a quick question about autism genetics research, and if any of you have ever participated in this. So I'm going to spend the next few minutes going over the details of our genetics research study. I'll let you answer the poll questions. And as I mentioned, we view this as a real partnership with the community and the families. We're grateful for their participation to advance the knowledge of this complex condition and ultimately our understanding of the human brain and its normal function. All right, so the vast majority of participants of our Science Cafe attendees have not participated in autism genetics research. So for our study, the eligibility criteria for participation is a clinical diagnosis of ASD. And individuals with one of the genetically defined syndromes, specifically Fragile X, Rett, Angelman, or TSC, are not eligible to participate in the study since the genetic causes of their symptoms are already identified. After enrollment of study participants, we collect blood samples from all available family members, and we extract the DNA and sequence the entire genome or exome of each sample. And exome just refers to the part of the genome that encodes for protein, or the coding part of the genome. These methods allow us to identify all types of mutations. And for families who are interested in participating in our autism genetics research study, they can contact us directly at our email address listed here, autismgenetics@utsouthwestern.edu. After we analyze the genomic sequencing data, there are three possible outcomes from the study. The first one is that we may identify benign variants. And these are just changes in the genome that do not cause any disease. They are normal variants in the genome that are present in the general population. And since this is not relevant to ASD, this result is not returned to participants. All right, and now you have an interesting poll question here. Do you think that 99% of our genomes are identical or not? And I'm gonna wait for this result to make a quick comment about it. All right. We're close to a 50-50 split. And the answer is that it's true. So 99.9% of our genomes, of all human genomes, are identical. And that 0.1% carries changes, or what we call genomic variants. And these changes can be responsible for traits like whether your hair is curly or straight, your eye color, whether you're tall or short, and other variants in the genome could be responsible for diseases or conditions that one may have. So as I mentioned, the first outcome from the study could be that we just simply identify benign variants. And in this case, we don't report these back to study participants because they are not relevant. The second possible outcome is that we actually identify variants that are of unknown clinical significance. So what that means is that at the time of the study, there was no information about what these variants do and whether or not they actually cause ASD. And because of that, we don't report this result back to study participants. The third possible outcome is we identify pathogenic variants. And these are variants that are known to cause ASD. In this case, we can return the results to study participants if they choose so. And I also wanted to add here that because of the amount of ASD research that's taking place and the wealth of data that's being generated by research groups worldwide, some of the variants that are initially classified as unknown here in our the second category that I told you about may be updated to become known pathogenic variants. So we periodically update our data, and if the classification of a specific variant changes, we will inform the participants. And now I just want to give you a quick glimpse of our ongoing projects and to summarize the autism genetics research study. So to date, we have enrolled over 3,200 individuals in our genetics study. And the cohort includes individuals along the spectrum of ASD and from many ancestors. This is an ongoing effort, and we've identified several new ASD genes so far. We are also in parallel using another approach to identify ASD genes, and this strategy is called forward genetics, and it's shown here in the bottom right corner. So in this strategy, mice are their mouse genome is mutagenized, and then we screen for what we refer to as ASD-like behaviors. Then we identify the genes that underlie these behaviors, and finally, we validate the relevance of our findings to humans. Importantly, after we identify ASD genes through these concerted efforts of human genetics research study and the forward genetics research in mice, what we're really interested in is to understand their normal function in the brain and to pinpoint specific mechanisms that they regulate. We're currently focusing on two genes. One of them is called KDM5A here in the top left. And the normal function of this gene is to bind to specific regions of the genome and suppress other target genes that it regulates. And by doing so, it regulates neuronal development in the brain. Another gene that we're focusing on is called UBE3B, as shown here on the right, top right corner. And this gene encodes for an enzyme that regulates the levels of many proteins, one of which that we've identified functions in regulating metabolic pathways in the brain. And this is an example of a pathway that we think might be targetable in the future, and we have ongoing studies to explore that option in mouse models. Our hope is that by studying these specific mechanisms that are affected in the brain when ASD genes are mutated, we will be able to pinpoint targetable pathways that we can then harness for the development of personalized therapies in the future. And in closing, I'd like to thank all of you and to thank the individuals with ASD and their families for participating in all of our studies and driving our research, the foundations and family support groups, and my lab members who do all the hard work, and the funding agencies that support our work. And we have a quick last questions about whether any of our attendees tonight has worked with advocacy or support groups. And with that, I will hand it over to Jenny. Thank you, everyone.
All right, this now Dr. Tsai, it's your turn. Thank you.
Awesome. Thank you so much, Maria. That's awesome. Just going to ask you to unshare. Let me share this. All right, so hopefully everybody can see that okay. So thank you so much, Maria, for, as always, a fascinating presentation. And thank you as well to Jenny and to the rest of the organizers for the opportunity to be here today and a huge thank you for all of you for showing up here in the middle of your busy week to kind of come and learn with us today. And you know, as Maria mentioned as well, to kind of the patients that we care for in the Center for Autism and Developmental Disabilities that really ultimately, you know, along with the autism community at writ large, kind of inspire us to do the work that we do. And so, you know, my title of my talk is Circuits in Autism, Potential Therapeutic Opportunities. You know, I think Dr. Cha Rohr really kind of, you know, kind of laid the groundwork nicely for kind of the talk that I'm going to talk about, you know, really detailing the genetic contribution that we see in autism, you know, detailing, you know, that there have now been over hundreds, if not thousands of genes that have been identified, but still not quite accounting for 50% of the entire autism pie, if you will. And so again, when all is kind of, you know, settled and when the, you know, dust settles ultimately, right, I think we're talking about probably thousands of different genes that are likely contributing kind of to the whole kind of spectrum that is autism. And so when we, you know, kind of when I was thinking about this, you know, as I was starting my lab and trying to understand, you know, clinically, how can we make the biggest difference for individuals with autism? You know, one of the big questions was, you know, there's a big challenge ahead of us. You know, if we go and we're trying to address individual genetic contributions, you know, we're going to talk about different, you know, thousands of different individual therapies, right? And so we wanted to ask, you know, whether there was a possibility to learn from what these genes are doing and try to understand, are there actually shared functions of these genes? You know, are the shared functions of these genes such that instead of developing a therapy that would target, you know, a 1% or a less than 1%, could we have and develop a kind of a clinical trial and develop a therapeutic for something that would maybe target 5%, maybe 10%, or potentially even more, right? And so it turns out that actually this is the case. That if you actually look at what these genes do, the vast majority of the genes actually have shared functions, and they have shared functions in how, especially in how nerve cells talk to one another. And that could be directly through these nerve cell connections that we call synapses. And that can be also through local connections with neurons that are right next to them, but also connections long range across the brain through long range connections that we would like to call circuits. So circuits really is just another way of saying kind of connections between different nerve cells, right? But ultimately, there's a problem here, right? Is that, you know, we have about 100, conservatively, maybe hundreds of billions of neurons or nerve cells within our brain. And each one is making maybe 100,000 connections to other nerve cells. And so, you know, if we are going to target specific circuits for potential benefit, you know, where do we start looking, right? I mean, this is a big, there's a big jumble of these neural circuits kind of within the brain, right? And another question that we have is, even if we could identify the precise neural connection that actually is really critical for the regulation of behaviors in autism, what can we do about it, right? And so those two big questions in some ways are big themes that we're trying to target in the lab. And so the first question I'm actually going to answer is actually the second question that I proposed. And one is that if you can understand the circuit, what can you do about it? So I use this very, very simple diagram of basically a fan and a light, right? And you can see, if you understand kind of what the wires do and what the components of this, you know, simple machine are, potentially if something goes awry, you can fix it. So if you go and you flip the switch, the light turns on, the fan turns on, you can regulate it through the little knob that we all have in our old school fans to take it on higher or lower. And so what happens when things go awry, right? So if we go and we flip the switch and say, now the fan goes on, but maybe the lamp doesn't turn on, right? Because we know this wiring diagram, and because we know the components of that wiring diagram, we can say, okay, well, it can't be the battery because the fan is still on. So it must be something within this local circuit here that's gone awry. Maybe the light bulb needs to be replaced. Maybe this switch broke, or maybe this wire got disconnected or something like that. We know because we know the roadmap in some ways, in some ways how to fix the specific problem that arises, right? Now, obviously, this is a very simple diagram, and the brain is a much more complex kind of beast, if you will, right? But could you imagine a scenario where if you understand the underlying circuitry and the nerve connections that are critical for, say, a social behavior or a behavioral flexibility, could we leverage that understanding to actually develop therapies that actually could improve and impact some of the challenges that our patients with autism in the autism community face? And this sounds a little bit sci-fi, if you will, right? Like, oh, we're going to do some Star Trek-y kind of modulation of the circuit, and that's going to improve behavior. But actually, it turns out that we have been doing this in neurology for quite some time. So if you, as an example from Parkinson's disorder, by understanding kind of the circuits that's really critical for kind of motor movements that are dysregulated in disorder, you can actually target the specific circuit and now actually have specific modalities that both impact and improve motor function, but also really, really improve the quality of life in patients with Parkinson's disorder. And so can we identify these circuits and potentially leverage these findings for individuals with autism? And so then it brings us back to this question of where do we start looking? And for me and for my lab, ultimately, the place to start looking came actually in clinic. And so when I was training, and so, by clinical hat, I am a child neurologist or a pediatric neurologist. And so during my training, there were a couple of really kind of formative, some ways formative interactions that I had with patients. And, these are just a few of them. But one of them was that in interactions with children with autism who presented to clinic, when I noticed that, in addition to obviously the core deficits that characterize this disorder, I also found that many of these individuals had a number of motor abnormalities as well. So motor challenges, right? Things like low tone, things like apraxia, some frank ataxia, and discoordination with their balance and with their movement. And interestingly enough, a number of these individuals with autism had received brain scans and were found to have altered structure in a part of the brain called the cerebellum that really traditionally has been thought of having a role specifically for motor control, right? And in addition, I also encountered a number of individuals who had primary cerebellar processes. And when I encountered them, thinking to myself, hey, the cerebellum is really critical for motor function, I thought to myself, hey, when I see these patients, the parents are gonna ask, hey, I'm interested in motor things that can improve their motor function, et cetera. And when I went in and saw these families and saw these individuals and saw these children, when I asked the parents, 1 through 5, what are the things that you were most concerned about? The motor and the movement had never made those top 5. It was all things like cognition, behavior, my ability to interact socially, my child's language, my child's kind of doing repetitive movements, these kinds of things, a lot of things that really were reminiscent of the behaviors that you see in individuals with autism. And it turned out around that time, a group at DC Children's, Dr. Limperopoulos and her colleagues published a number of really seminal papers describing the prevalence of autism and neurodevelopmental challenges in individuals with primary cerebellar processes, if you will. And so with all this, we wanted to ask, is there a contribution of this part of the brain, the cerebellum, in the regulation of these behaviors? And so, I threw up this question. I think there's a poll question that's going to pop up related to this. I think it will pop up here. And so one of the questions is, how similar are we to mice? And the reason I asked this question is because, when we're thinking about models, how do we model and ask this question of whether cerebellar dysfunction is critical for the regulation of these behaviors? We decided ultimately to use rodent models, as I think Dr. Chahrour mentioned in her talk as well. And it turns out that when we disrupted a gene that has been linked to autism, specifically only in the cerebellum, we found that these animals demonstrated a lot of the behaviors that are reminiscent of the behaviors relevant to autism: social impairments, repetitive behaviors, behavioral inflexibility, alterations in their communication, etc. And so it looked like the cerebellum appeared to have a critical role in the regulation of some of these behaviors. So we then wanted to ask, okay, the cerebellum's a big place. And in fact, a random trivia fact about the brain is that actually about 70% of all of your neurons exist within this part of the brain, the cerebellum. And so we wanted to ask, can we drill down a little further? Actually, can we identify which regions within the cerebellum actually are critical for the regulation of these important behaviors? And so we went and we looked, and we first adopted a kind of imaging-based approach. We took pictures using magnetic resonance imaging, or an MRI, that many of you or your family members might have taken. And it turns out in this particular mouse model, we found regions of the cerebellum that were specifically structurally altered. And it turns out some of these regions actually are those same regions that, if you do these studies in individuals with autism, you see altered structure within these particular regions of the cerebellum. And so we wanted to ask, are these regions critical for the regulation of the behaviors? And so we used a number of different rodent models as well, showing that in fact, disruption of these specific regions are important for the regulation of both social behaviors as well as behavioral flexibility, right? And so we then wanted to ask, well, that's great if we can identify these regions, but we also know from the whole literature about how the cerebellum works, the cerebellum does not act alone. It is not a lone ranger, if you will. It acts through communications networks across the brain. And so we wanted to ask, can we identify those connections or circuits, as I mentioned before, that are being used and interact with the cerebellum for regulating these important behaviors. And it turns out in these animal models, we found alterations in those connections or in those neural circuits to regions of the cerebral cortex that have also prior been implicated in individuals with autism. And so we then wanted to ask, well, that's great. So the anatomy, these connections are there, right? But we also wanted to ask, are these connections actually being used to talk to one another, right? Or is it just that these connections, these alterations of these connections are just kind of an artifact, or in some ways a red herring, and are completely unrelated potentially to autism and the behaviors that we're interested in, right? And so it turns out we're able to show through a number of different studies that in fact these regions are talking to one another, and importantly, that these neural connections are actually critical for the regulation of these behaviors that are relevant to the individuals that we care for in clinic: social behaviors, behavioral inflexibility, et cetera. And in understanding these connections, if we go back to those mouse models of where we disrupted specific genes that have been linked to autism, we can leverage and modulate those circuits and actually impact those social behaviors and behavioral inflexibility that are disrupted in these models, right? And so that's great. So we understand the mouse a little bit more, right? And that's wonderful, but at the end of the day, our goal is not to understand the mouse, right? And we want to know, even though, I showed before in this poll, that mice actually have, really significant overlap in identity to humans, and actually it's much greater than 80% was the answer to that poll. And in fact, there are many different genes that actually have over 99% identity, many of which actually have been linked to autism. And so we wanted to ask this question, right? Is this actually also relevant to humans, and importantly to individuals with autism? And it turns out that actually that is the case, that we see those circuits that are disrupted in our mouse models of these autism genes, they're actually also disrupted in large cohorts of individuals with autism. And it turns out that these regions of the brain in humans are also talking to one another in the same way that these regions seem to be talking to one another in animals, in our rodent models. And so we wanted to ask this question ultimately, right? That we have shown that there are regions in these mouse models that seem to have critical roles in the regulation of these behaviors. These regions have been implicated in individuals with autism. We see disruptions and connections both in our mouse models of autism-linked genes as well as in individuals with autism. We see that these regions are talking to one another both in these rodent models and in individuals with autism. And we see that if you modulate these circuits, potentially you have the potential of impacting behaviors that are relevant to autism, at least in these animal models. And so we wanted to ask this question, and we have now ongoing clinical studies to ask whether these circuits are involved in individuals with autism, and furthermore, whether modulation of these circuits might actually impact and improve behaviors in individuals who are seeking help for the challenges that they face kind of relevant to autism, whether that be in the social domains or in kind of that behavioral flexibility domains. And we have these ongoing studies. If anybody's interested, feel free to reach out. We have this email, autism@utsouthwestern.edu, that we can put into the chat if anybody's interested in finding out more. We're more than happy to talk kind of after this. So all that to say, I just want to say a huge thank you again for your attention, a huge thank you for the organizers for allowing me to participate here today. And I think after this, Dr. Cha Rohr and I would be more than happy to take any questions.
Thank you, Drs. Cha Rohr and Dr. Tsai, for your presentations this evening. We will now get into the Q&A portion of this session. First, we'll start with some questions submitted ahead of time, and then we'll address questions from the chat if we have any. We received several questions about diet and nutrition. How can diet impact autism diagnosis and care? Is there a more favorable diet that for those with autism? And what about the link between vitamin D deficiency and autism? And Dr. Tsai, do you want to take that one?
Sure. So there was a lot of questions in there. And so I will try to, I'll try to address them, and please tell me if I forgot any, and I will try to address them. So maybe since it's the freshest in my mind, we'll talk about vitamin D first. And so there have definitely been a lot of discussions in the media about, is there a link between low vitamin D specifically and a risk or increased risk of developing autism? And the short answer is that the jury's still out. I think is what the short answer. Now, to elaborate on that a little bit, so there have been many studies taking a look at this, both in kind of animal models as well as more population-based studies, linking low vitamin D levels with increased risk. There, however, have been a lot of kind of differing messages, if you will, some concluding that where the low vitamin D relevance is actually low vitamin D in pregnant moms. But then there are other studies that have looked and said, actually, it's not the pregnant mom, actually it's in the newborn infants who ultimately then emerge with a diagnosis of autism, right? And then there's some studies that actually show that it's not one or the other, it's actually you need both to be low in order to increase your risk, right? And then there are, other studies that have shown, hey, if you do vitamin D supplementation, that actually can improve outcomes and improve, if you do kind of standardized testing for autism relevant behaviors, that maybe things will improve. The flip side is that there are also equal number of studies that show kind of the opposite, that actually there doesn't seem to be a connection between vitamin D and increased risk. There are also a number of studies that show that even with significant vitamin D supplementation, that there does not appear to they do not see benefit for individuals. And so as a result, many of these studies are with small sample sizes. And so I think ultimately, when folks have actually went and looked at this literature and reviewed it, the ultimate conclusion is that we don't know yet. There may be a link, and that ultimately more and better and larger studies with larger cohorts of individuals with autism participants are going to be needed to really best answer this question. So that's the vitamin D aspect. I think now in terms of diet, I think it's it's very clear that we see, in the clinic that many of the individuals with autism have specific dietary preferences. So there's actually this entity called ARFID, which is this long name of avoidant restrictive food intake disorder, I believe is what it is, ARFID, that is present in many individuals with autism. And again, it's restrictive eating that folks can represent. And sometimes when you think about restrictive eating, you think, oh, they're not going to be eating enough, you need more calories and this kind of thing. But actually it turns out that a lot of these individuals, because they have restrictive food intake, they are very restricted to specific types of food. So, hey, I will only eat French fries, or I will only eat peanut butter. And so it's not that they actually have low calories, that some kids actually have the opposite, where there's too many calories. They're just specific types of calories that, that ultimately result in, sometimes metabolic disturbance, not getting enough of the full array of minerals and nutrition that one needs. And so you can get kind of just like individuals with autism, there's a huge spectrum. There's also a huge spectrum in terms of dietary needs and challenges, if you will. And so as a result, we, for most of these kids, we strongly, I kind of would advocate for working closely with a nutritionist, because there are a lot of things that can be done to really try to diversify the diet. Because I think, as we have learned kind of going through, in not just with autism, but I think kind of in the population at large, having a nice varied diet when you are moderate intake of all of the different types of food groups is really the best pathway to health. And I think that is also ultimately true for our patients and for individuals with autism.
Thank you, Dr. Tsai. Next, we have a question for Dr. Cha Rohr involving forward genetics. Can you expand on how this research technique is helping you in your lab discover more about autism and how to improve diagnosis of autism? Are there many common genes you are finding that relate to autism?
Yeah, thank you for that question. So the forward genetic screening is the second strategy that I spoke about on how we identify autism genes. So basically the idea here is that this strategy allows us to be much, much more efficient and to do this in a high throughput manner. So we can, instead of the traditional genetic studies where we start with our phenotype or trait or the condition that we're studying, and then we sequence the DNA to look for what potential mutations could be causing this, we do the opposite. So we go ahead and we induce these mutations in the genome. So we introduce random mutations across the mouse genome, and then we ask, which of these mutations that we introduced gave rise to behaviors in the mice that may indicate abnormal brain development? So it's an unbiased strategy. It's very high throughput. We can do this at a very large scale. And at some point in time, as we continue doing this, we'll be able to saturate the entire mouse genome with mutations. So this is a much, much faster strategy than if we went out and looked for mutations in the human population. And that's what we do on a daily basis. But the idea is that having those side by side, being able to do this much faster method in the mice, and then go back and look, is there a relevance for these genes that we found in mice to humans? And at the same time, we're continuing our human genetics study and identifying genes through that method as well. So the two approaches are complementary.
Thank you. Dr. Cha Rohr, your slides mention autism is found in males more than females, but there seems to be more women getting diagnosed as adults. Does the genetic data reflect the underdiagnosis of girls?
So this bullet point about the higher prevalence in males, about 3 times more than in females, that actually comes from prevalence studies in the population. And it does reflect an underdiagnosis in females. It reflects multiple things. So there's definitely an underdiagnosis in females because a lot of the initial diagnostic methods and tools were more geared toward males. So that's one. And the other thing is that there are some mutations that are on the X chromosome, and so they tend to affect males more severely. There's also, other hypotheses out there for why this might be the case. It's still, a lot of research is ongoing about that, but that definitely there is an underdiagnosis in females, and that's kind of catching up now. And that's why the number is about 3 times rather than 4 times or 5 times higher. Yeah. Thank you for that question.
Okay, our next question is, does exercise reduce severity? Does a pattern exercise regimen help more? And I'll leave this for either one of you to answer.
So I do know of one example in a study that was done on Rett syndrome, which is a very severe form of autism. It was done in a mouse model of Rett syndrome, and it showed that a regimented exercise program actually improves a process called neurogenesis, which is the birth of neurons in the hippocampus, which is a region of the brain that controls learning and memory and other important processes. And so this study showed that a regimented exercise program actually improved some of these autism-like behaviors in a mouse model of Rett syndrome. But it's still a study in a mouse model, but it's very interesting, definitely. Yeah.
Thank you. Dr. Tsai, many people, this is a question from the audience, many people like my child are not diagnosed until they are adults. Are there studies about autistic adults?
Yeah, so this is a great question. And so the short answer is there needs to be more. There are definitely studies that are ongoing in adults, both I think from a diagnostic standpoint, from a mechanistic standpoint, and trying to understand, kind of causes, but also from a treatment standpoint. But as I think the question alludes to, there are many, many more studies that have focused on children. And traditionally, I think the focus has been there. I think it's very, very clear that, kids become adults, and that we need to understand better the challenges that individuals with autism face as they get older. And also, I think, not just the kind of the challenges, but also whether there are different therapeutic opportunities, if you will, that may be different in children and for adults. And those things definitely need to be studied more. And it definitely, I think there is an increase in interest, and I think it's very clear that there needs to be even a greater interest. And I think that is those ideas are changing, and hopefully it will continue to change as time goes on.
And I just want to add one quick comment that our autism genetics research study is open to individuals with autism of any age. And also that's one of the things that we work towards is, to improve our understanding of all of these genes that underlie autism so that we can improve genetic diagnosis, which could be done much, much earlier than a clinical diagnosis. And we hope that in the future, access to diagnosis will be available to everyone more readily, I should say.
Yeah, just to hop onto that as well, I would say the same for our studies as well, that we have studies that are focusing on children. We're actually doing kind of similar studies as well with our partners for adults with autism as well.
Here's another question from our audience. I have read that autism risk increase in the age of the father. Are there factors that seem to increase the incidences of autism? Do you think we're seeing an increase due to the use of pesticides, fertilizers, et cetera? And might that show up in that might show up in our food or maybe pollutants in the air or water?
So I'm gonna take at least the first part of this about the age of the father. So there are a lot of studies that have looked at this and found that there is an increase in the risk of autism in offspring from older fathers. And one of the reasons why this might be the case is that with age, the sperm from males accumulates more of these de novo or spontaneous mutations that I talked about. And so that increases the risk in the offspring. But also there's a distinction between something increasing the risk versus something being causative. An increase in risk just means that your risk is higher, but that doesn't necessarily mean that you will actually end up developing a particular condition. So there's a distinction between an increase in risk versus a causative or something being causative. In terms of other environmental factors, and age of the father is definitely one environmental factor. Other environmental factors there is no strong evidence to support any of these. Yeah, so Peter, I don't know if you want to add anything about those.
Yeah, I mean, I think a little bit like the vitamin D story. I mean, the vitamin D story tends to have actually a little bit more data supporting, and it's still, I think, still inconclusive. But I think for a lot of these environmental factors, whether they be pesticides, various kind of components within the diet, et cetera, really the jury is still out. I think these things need to be studied more because potentially there are contributions, but I think the jury is very much still out.
Thank you. Dr. Tsai, how frequent is autism in siblings and generationally in families? Is autism often misdiagnosed in early childhood?
Yeah, so I mean, this is actually a great question for Maria too. So the short answer is if one has autism, the risk of a subsequent future sibling, et cetera, having autism definitely go up. And so I think if you look at the rates of autism in an identical twin, for instance, you're talking on the order of 80 to 90% of developing receiving that diagnosis. If it's a fraternal twin, you're probably on the order of 20% or so, high 20s kind of thing. And if it's a sibling, but not a twin per se, probably in the higher teens, again, as opposed to, I think Dr. Chahrour mentioned in her slides, a population risk of about 2%. So definitely an increase because of the genetic factors that presumably a sibling shares with each other.
Yeah. So I'm going to comment on that real quick too. So this, as I mentioned, autism is highly heritable. And actually it's through all of these twin studies and sibling studies, that's how, we figured out that autism is highly heritable. And as Peter mentioned, the more you share of your genome, the higher the risk is. So if you're, monozygotic or identical twin versus fraternal twins versus siblings, your risk is higher as identical twins and so on.
Thank you. Any evidence to support ABA therapy? And I'll open that for either one of you to answer.
Yeah, no, I'm happy to answer. I mean, I think so. The data is actually very, very clear in terms of benefits for ABA. So, if you think, I think someone in the chat had asked about what therapies are available. Unfortunately, I wish, and I think Dr. Chahrour would echo this, that we wish there were more therapies for the autism community. What I would say is the data that we have best in terms of best kind of therapeutic kind of contribution, if you will, right? The biggest impact is actually data supporting ABA, and that's on the order of 50% of folks who receive kind of really intensive ABA have a significant reduction in the need for special education services. Now, if you look at kind of the fine detail, of course, is that the amount of ABA therapy that one needs actually on the order of 20 plus hours of therapy a week. So a significant amount that unfortunately to most individuals, definitely in the state of Texas, but across the nation, they're just not able to kind of receive for insurance reasons, for availability reasons, and specifically in the state of Texas, if you look at the number of ABA trained providers, just to give a couple hours of ABA therapy a week for individuals with autism, we have a deficit of providers greater than tenfold, right? And then that's across the state. Now, if you are, in a small town in West Texas or somewhere in the Panhandle, for instance, where maybe access to those providers is even less, it's really, really difficult to achieve those services, unfortunately. But the data is, I think, if there is, for all of the therapies that are available, in our quiver, if you will, ABA has the best data to support it. Now, I think there's lots of data. I think there was a question that someone had submitted in terms of could ABA itself be detrimental? And there was a study, I think, I want to say, and again, don't quote me if I'm wrong, but I think it's in 2018. I could be getting the years wrong, where there was noted a potential association between ABA and PTSD. But actually, when you really look at the study and how it was done, it's interesting. This study was done as the researcher sent out questionnaires to maybe, I think, 200 or so folks with adults, and 200 or so children. And very few responses came back. Less than 50% of things came back. There was no standardized testing of people diagnosis with autism or diagnosis of PTSD. So it was all self-report, saying I'm an individual who has an autism diagnosis. I'm a person who has PTSD symptoms. And so there was this link. And then in addition, if you look at actually the responders of that population, so again, only half responded, and only really about 10 people actually in the adult population actually had some evidence or some self-report of ABA. So we're really talking about 10 individuals of folks that were diagnosed in adulthood with autism, but had some recollection of receiving ABA when they were children. And so there are lots of questions about, did they receive, did they truly receive kind of an autism targeted ABA? What kind of, and what was the quality of that ABA? What was that ABA for? And if you look at population in general, is it possible that actually just the symptomatology that is associated with PTSD, significant anxiety, some sensory abnormalities that folks with autism can also have, are they actually confounded on one another, and is it truly a PTSD diagnosis? And so with that, all that to say, I think the data is very, very inconclusive. And so, I think, again, what I would say is the data for ABA and the benefits of ABA are very clear. And, again, I think potential side effects in all of these kind of studies, these long range studies, need to be done. But I think right now the evidence is much more in favor of benefits as opposed to detriment.
Thank you. Is there a connection between ASD and OCD?
So OCD is one of those comorbidities or conditions that can co-occur with ASD, and actually up to 24% of individuals with autism also have OCD. Yeah.
And interestingly, from a mechanistic standpoint, right, if you do imaging, so brain imaging studies of individuals with OCD versus looking at brain scans of individuals with ASD, actually, there are multiple studies that kind of show that they actually look very, very similar. And some of the brain circuits that are kind of disrupted in one are often seen also in the other.
Thank you. The next question is, what is the difference between multiple symptoms of disabilities and actually being tested? And what are the qualifications of this process to happen or get set up?
Sorry, I missed the question.
Okay, let me repeat it. What is the difference between multiple symptoms of disabilities and actually being tested? And then what are the qualifications for this process to happen or to get set up? We can come back to that one. This is a question another question from the audience. Is there any connection to autistic symptoms and how one gets autism?
So, I think it's a great question, and I think they're in some ways, we're still at the stage of, I think as Dr. Chahrour and kind of my talks, in some ways express, is trying to actually still understand what are the causes of autism. And because, you meet one individual with autism, you met one individual with autism, there's a huge heterogeneity and a huge spectrum that makes up the total autism, population, if you will, right? And so what causes one versus the other? Is it the same? Is it the same gene? Is it a different gene? Are there environmental factors that contribute? And, I think it's ultimately gonna, when the dust settles, I think it's gonna be a very complex intermix between kind of environment and genetics. And so right now, because of that, I think the jury really much, I think we just do not know specific, for each individual, what is your specific cause for autism in most of the individuals that we see. And so as a result, it's difficult in some ways to say, well, because we don't know the genetic, we don't know necessarily the specific cause for your autism. How does that ultimately relate to the specific phenotype that you're expressing, whether it be, are there comorbidities of anxiety, how significant your social impairments are, et cetera. And so those studies, without a doubt, really need to be done, and probably done kind of on a really large scale. So a lot of the scale that Dr. Chahrour is mentioning in terms of genetic studies, pairing that with kind of really deep phenotyping in terms of, hey, these are the challenges that this individual who has this genetic alteration has, and doing that at large scale to really start to get at those important questions.
Thank you so much. Unfortunately, we are out of time, but I know that we could go on, so we'll have to have you all back. Thank you very much, Dr. Cha Rohr and Dr. Tsai, for providing insight into your work. It's important research and treatments for these precious children and advocating for them and their families. So thank you. Thanks also to Charlie, Amira Wynn, Holly Norris, Melissa Martin, Diane McGee for helping run tonight's event. You guys were really helpful. We do have a slight change from our regular bi-weekly schedule of science cafes. This coming Monday at 12 noon during your lunch hour, we are hosting a special Zoom conversation about mental health counseling, our pediatrics residency, and the nonprofit Cafe Momentum that many of you might know about in Dallas. It works with kids out of the juvenile justice system. After that, the next Science Cafe regarding the science of coughing will be on Thursday, April 14th. Charlie is putting registration links in the chat, and we will also email you with this information. We just want to thank everyone again for joining us on behalf of our faculty, speakers, and the Science Cafe team. As we close out this evening, we wish you all good health and wellness and a good night. We are adjourned. Thank you.